All right, Catherine, take it away.
Thank you. Thanks. So for all those who don't know, my name is Catherine Friend, the Director of Chameleon Science, and we're very, very proud to be the Australian distributors of the Fluent BioSciences product range. Today, I'm really, really pleased to have with us the senior scientist and head business development manager of Fluent BioSciences, Jessie Zhang. Jessie, thanks very much for joining us, and probably just going to pass it on to you to just start the webinar. If anyone's got any questions, please put them into the chat or the Q&A. I'll probably stop Jessie in the middle of the talk and just ask a few questions. Feel free to put them out throughout the talk, or we'll have some time at the end as well to run through a few questions if you have any further ones. Without further ado, I'll leave it up to you, Jessie. Thanks very much.
Perfect. Let me share my screen and let me know if you have any trouble seeing the screen. So let me. Okay, so I'm excited to announce kind of the launch of Fluent BioSciences PIPseq product line in Australia. And this has been facilitated through working with Catherine and Chameleon Science. Been great partners to work with thus far and bring the product over to Australia. Really, the company mission of Fluent BioSciences is to bring single cell analysis into any lab, really to democratize the space. We've been commercial for about 2 years with our PIPseq product line, and it's based around a 3' single cell RNA sequencing platform. One of the biggest advantages of PIPseq, and the reason why we're able to advertise it as being so easily able to set up in any lab, is that it's powered by this particle-templated instant partitioning technology, which I'm excited to talk to you more about in the coming slides. If you're interested in further reading, our paper describing the technology was published in Nature Biotech about a year ago. If you do look at that paper, just keep in mind that it's actually referencing an older version of the chemistry. The version that I'll be focusing on today is the V4 Plus version of the chemistry. In addition, we've also had several other preprints as well as peer-reviewed publications from other users that have successfully adapted PIPseq for their workflows that you also may take a look at. And if you go to our webpage, www.fluentbio.com, there's actually a publications page where you can you have easy access to all these publications for further reading. So the advantage behind PIPseq is that we're really trying to eliminate some of the barriers to setting up single cell analyses in various settings. And in doing our market research, some of the constraints that Fluent identified in the single cell RNA sequencing space is the requirement for expensive instrumentation and consumables, solutions that were either limited in scale and or had rigid workflows. Finally, there's also a significant expertise barrier to be able to successfully perform a single cell assay. And PIPseq aims to address all of these limitations by offering a solution that is highly scalable, the same workflow, and the same benchtop equipment can be used to process any number of cells ranging from hundreds to hundreds of thousands. The assay is also highly flexible, having been successfully demonstrated on a variety of different sample types from different organisms, including cells and also nuclei, as well as other multi-omics add-ons to the core single cell sequencing assay. Our solution is also highly affordable. Cost per cell is about half or less of alternative platforms out on the market, and that figure only gets better as you scale to our larger input kit sizes. Finally, the workflow is quite simple, taking about slightly under 2 days of hands-on time in the lab. And there also are many flexible stopping points built into the assay, and I will highlight a particular one that is quite unique to relative to other platforms on the market, and many of our customers have chosen to take advantage of. So as I described earlier, the core innovation behind PIPseq, the reason why we're able to bring highly scalable single-cell RNA sequencing to the benchtop, is through this technology that generates monodisperse emulsions via vortexing. The way this technology works is that we provide you with a tube of barcoded hydrogel beads to which you add your single-cell suspension and partitioning fluid. And by placing that mixture onto our vortexer and simply vortexing for 2 minutes, the resulting output is a highly monodisperse emulsion that looks like it came out of a microfluidic chip, except that it doesn't require any microfluidics, just a vortexer. And the great thing about this process is that it scales very nicely to any tube size. So we successfully demonstrated this vortexing emulsion generation procedure on tube sizes ranging from PCR tubes to 50 ml conicals. And as you can imagine, this allows us to scale for different larger or smaller input sizes just simply by changing the geometry of the tubes, by changing the size and also the number of pips that we have in each tube. And all of this process happens within minutes. And for those of you that are not that are new to the single cell space, the act of vortexing and generating this droplet is what captures your single cell. This allows you to get extremely efficient single cell capture. Now jumping into a little more details about the actual workflow behind PIPseq. As I mentioned earlier, the vortexing procedure is what captures your single cells and co-localizes them with the mRNA capture beads. After that process, you will then place the sample into into a dry bath, basically to start the thermal-based lysis and mRNA capture. This is actually one of the first stopping points of many in the assay, and one that's actually kind of highly unusual for the field, for the space. The emulsion at this point is actually stable for at least 96 hours at room temperature, which is quite remarkable, given that in this state, you're basically working with naked mRNA that is captured onto the beads. However, we found that this stopping point has no impact on data quality, and this is great because this is less than 10 minutes of hands-on time into the workflow. So if you have a very long sample prep, you don't need to go very far into the PIPseq workflow before you can reset and come back the next day or the next, after the weekend's over, and attack the workflow with fresh hands. After lysis and mRNA capture, the captured mRNA is reverse transcribed and then PCR amplified, following which the cDNA is taken through a very standard fragmentation and ligation-based library preparation and preparation for sequencing on Illumina machines. After sequencing, we offer a free to download and install bioinformatics software package called PIPseq that enables effortless first looks at the sequencing performance, as well as providing additional outputs for more sophisticated tertiary analyses. And I'll talk a little bit more about PIPseq on the next slide. So PIPseq is the PIPseq analysis pipeline. And as I mentioned before, it's free to download and install, and it works on all major operating systems. In addition, for those users that may not have the computing power necessary to process the pipeline, we also have partnered with several cloud-based service providers that have installed PIPseq on their infrastructure. This includes BasePair and LatchBio. So just simply by creating an account on those web apps, you may access the PIPseq software and be able to process your sample all without having access to powerful computing clusters. What PIPseq allows is it has many different features that are associated with bioinformatic analyses of single cell data. But at the very core of it, with every single PIPseq run, you will get a report that lists key performance metrics from your sequencing run, as well as some first glances into the clustering of your samples, as well as genes that are associated with each cluster. So even without diving into tertiary analyses, you can already start to get a flavor for how your sample performed how your single cell experiment performed. Another feature that's unique to PIPseq that quite a few of our customers have appreciated is the ability to adjust the cell calling, basically tuning the sensitivity of the cell calling to either include or exclude more cells. And the advantage here is that you get all of this information as well as the gene expression matrix files that you can take into your own tertiary analyses after you have viewed this report. Lastly, I also want to add that the PIPseq software is natively compatible with multi-omics approaches that are associated with 3' single-cell RNA sequencing. This includes approaches such as sample multiplexing via cell hashing, as well as antibody oligo labeling, as well as others. Some basic specifications about PIPseq are shown on the slide. When processing a human-mouse cell line mixture through our kits, one can easily resolve different, clearly distinct populations of mouse and human cells, indicating that our assay is highly single-cell concordant. In addition, replicates of the same sample type processed through different PIPseq reactions on different days yield highly correlated gene expression, indicating high reproducibility. This is actually one of the advantages of PIPseq that has allowed, in addition to the cheaper cost per cell, has allowed some of our customers to consider running technical replicates, whereas in the past or with other platforms, they would only have had the budget to run one single cell sequencing experiment. Finally, when comparing the gene expression concordance with other single cell platforms, we also find that we have a very high R value. We currently offer 3 different kit sizes, and they're named after the number of output cells that each kit is designed for. Namely, we offer the T2, T20, and T100 kit sizes. Each kit comes with multiple reactions per kit, and all of them have a similarly high capture efficiency owing to the way in which we perform single cell capture. One other thing of note is that we do not have a minimum cell loading requirement as well. So this capture rate is scalable regardless of how many cells you load into the kit. Our current chemistry is the V4 plus chemistry, and compared to the previous version of the chemistry, the V3 chemistry, we've made further improvements to the cell capture, sequencing efficiency, as well as the genes and transcripts per cell. We've also added compatibility with nuclei and cell fixation, as well as an overall improved user experience. This slide highlights some of the comparison data that we have between us and 10x done on the same lot of PBMCs. What I want to highlight is that in addition to PIPseq performing similarly across all the different cell lines, cell types within the PBMC sample, I like to highlight that for about the same price, one can obtain almost 10 times as many cells through one reaction of PIPseq as they would obtain through one lane of 10x chromium. Furthermore, when you look deeper into the correlation between the gene expression across all the different cell types in this PBMC sample, you can find that we are, again, highly concordant with 10x, meaning that those of you who have previously analyzed samples with other single cell platforms should find it totally acceptable to be able to integrate new data acquired with PIPseq into existing studies. I want to highlight that we are also compatible with nuclei prepared from fresh frozen tissue. These are just some clustering plots showing our performance with extracted nuclei from various mouse tissues, again, showing the great diversity of cell types that we were able to obtain. As mentioned earlier, we also are compatible with fixation-based approaches. Shown here on this slide is our performance with fixed nuclei, and what we found is that nuclei, when after having undergone fixation, this is with a DSP and methanol-based fixative maintain high quality gene expression profiles for up to 2 months post-fixation. Next few slides are just highlight some of the different applications that some of our customers have used across the different kit sizes. The T2 kit is a great starting point for both pilot studies as well as providing experimental flexibility in multifactorial studies. In the leftmost study, they took advantage of these researchers took advantage of the stopping point about 10 minutes of time into the assay that I mentioned earlier, and they used that took advantage of that stopping point to perform a time course study. So what they did is they processed different samples at different time points up until that first stopping point, and then when the last sample was processed, they then took all the samples together and processed the rest of them in parallel through the end of the workflow. The middle study is a study that involved many different clinical samples with different conditions. Finally, the last study on the right is actually an example of truly highlighting the accessibility of PIPseq, as this data shown on the slide was the result of establishing PIPseq in a community science center. So these were just regular adults from the high school students from the community coming to this facility to perform PIPseq. It really highlights again the kind of the portability and ease of setup of this platform. The T20 scale has been very popular with our customers to really begin to interrogate cellular diversity in complex tissues. For example, in the context of immunology, we've had customers identify exceptionally rare cell types in their samples at the abundance of 1% or less. We've also been able to, we've also had applications of the T20 being used to really nicely interrogate cellular diversity across a variety of different clinically relevant tissue, such as breast cancer sample in the middle panel, as well as mouse brain, whole mouse brain in the right panel. As brains are one of the more complex tissue types out there, this customer also ran the T100 on the sample and really was able to get a really nice looking clustering plot resolving many different tissue types from this complex tissue. And one thing I want to highlight for any neuroscience researchers that may be on the seminar today is that one of the biggest advantages of PIPseq relative to some of the other platforms out there is its ability to tolerate certain levels of non-cellular debris and sample prep. Thus, with PIPseq, preps that would maybe of concern in other platforms, we've actually had quite a bit of success with in PIPseq. And a great example of this is neural tissue, which actually contains large amounts of non-cellular fatty debris that has been problematic in some other platforms. Earlier, I mentioned that the PIPseeker software is compatible with multi-omics approaches that use 3' single-cell RNA sequencing as a base. To enable the generation of these libraries on the wet lab side, we offer an add-on kit called the C-Kit, or the Supplemental Enrichment and Amplification Kit, that enables such applications. These can include applications of a separate cell-associated synthetic nucleotide tag library in addition to your main gene expression library, enrichment of guide RNAs, for example, in CropSeq-like applications, as well as some more interesting kind of custom workflows that we have really enabled our customers to be able to do. And the key innovation that enables this is that our templates remain bound to the bead throughout the whole workflow, meaning that you can PCR amplify using the bead-bound reverse transcribed template with one set of primers, wash the beads, and then run a second PCR with a different set of primers, say a set of primers that targets, specifically dials out or amplifies the synthetic nucleotide library, or let's say you're interested in a small panel of genes such as transcription factors that you want to selectively amplify out. This ability to perform multiple PCR amplifications with different primer sets on the same template is really what one of the unique selling points of PIPseq. And what this all culminates in is the generation of parallel libraries that can be processed to generate multi-omics single cell data. The seq-kit provides reagents that provides PIPseq validated reagents that will enable this workflow, these sorts of workflows. This is just showing some data with our showing our performance using the reagents in the seq-kit to perform fairly common multi-omics assays. So we have a demonstration of CiteSeq done on PBMCs. So this is labeling your sample with antibody conjugated oligos. A similar sort of strategy can also be used for sample multiplexing. So on the top right of this panel, we have a demonstration using the seq-kit to do a cell hashing based experiment showing very good separation of the different hashtag groups. As mentioned earlier, we also are compatible with through the seq-kit workflow, CropSeq. So this is data actually taken from our Nature Biotech paper that shows our performance on a CropSeq library consisting of about 150 different guide RNAs. So this really, one of the advantages of PIPseq is you really can take advantage of the scalability of PIPseq to very cost effectively do large scale pooled genetic perturbation screens. So just to take, switch gears a little bit to think a little bit more about forward-looking compatibility. Sure, as all of you may be aware, there are going to be many different players coming onto the NGS instrumentation side. And we've actually had successful demonstrations with quite a few of the major up-and-coming players out there. So this includes Element, Instrument, Singular, and Ultima. You've also had demonstrated compatibility with the complete genomics sequencing instrument. And generally, all of these platforms will sell, although our library kits are designed to produce a library that's compatible with an Illumina instrument, all these companies sell kits that allow you to take a Illumina sequencing library and convert it to one that is compatible to their machine. So this should not be something that should hold you back from running your PIPseq experiment. Finally, we've also had a number of collaborations with researchers interested in long-read sequencing. So this both includes using the PacBio as well as the Oxford Nanopore instrument. Oh, is this the... Okay. So just to conclude, PIPseq really enables single cell analysis in every lab. And just to highlight some of the major, advantages of PIPseq, it really allows you to set up single cell RNA sequencing platforms with no capital investment. It's highly cost effective and accessible, really bringing the ability for you to perform these workflows to your benchtop in the lab. It's highly scalable from hundreds to hundreds of thousands of cells. We are working on a eventual million cell kit as well. We're compatible with a variety of different sample types, including cells, nuclei, fixation, as well as fungi and plants. We have intrinsic compatibility with all the different multi-omics assays, and our platform is also highly positioned for a clinic and point of sample collection. Had a few customers run, for example, this workflow in sort of BSL-3 environments where it would be cost prohibitive otherwise. And the table on the right's a little bit outdated because I realized that this is actually a slide from last year. But overall, this table highlights the major products in our product portfolio: the T2, the T20, and T100 kits, as well as the add-on kits such as the SEA kit, a 96-plex unique dual indexing kit, as well as the nuclei isolation kit. Finally, the cost to start up PIPseq, as I mentioned earlier, is fairly nominal. It just consists of two pieces of equipment that fit onto your benchtop. This includes both the vortexer and the dry bath. Both of these pieces of equipment have been highly customized and engineered to really enable reproducible performance. And the great thing is that once you buy a piece of starter equipment, you can actually use it for all the different kit sizes across our portfolio. In addition, there may be other uses for the instrument as well that you can experiment with once you have it in your lab. So with that, before I close, I also like to just give the room a little flavor of what's coming up in the product development pipeline. Right now our current chemistry is the V4 Plus, and sometime later in the summer, we actually anticipate releasing the V5 chemistry, which keeps all the advantages of the V4 workflow, but simply we've made improvements to the cell capture, the sensitivity, so the genes and transcripts per cell. So you should expect improved capture rates and about 30 to 60% improved genes and transcripts per cell. In addition, another project that, or another kit that we are actively working on is our direct capture CRISPR sequencing kit. So this is a kit that will enable users to have pooled screens that utilize Cas9, CRISPR-Cas9 based gene editing to be able to process those screens through single cell analysis without having to make a modified vector that fits the constraints of PropSeq. So with that, I will close. Happy to take any questions. And if you think of any questions after the seminar ends, feel free to also reach out to Catherine as well. Thank you.
Thanks very much, Jesse. May I just point out, though, guys, the pricing in here that was presented in his presentation is in US dollars. It's different pricing in Australian dollars, of course, because of exchange rates. So I'm happy to give you kind of a general idea of pricing as well, if you're interested in that as well. So just thought I'd point that out in case you get a bit of a shock. We send an email out to you for the quotes. In the meantime, though, I'd like to open the floor up and see if anyone's got any questions. Feel free to put it into the chat or the Q&A. I'm happy to kind of have the questions come through. In the meantime, actually, Jesse, I've got a question. So you said it's compatible with other multi-omics platforms. What platforms have you tested it on and what doesn't work?
Yeah, so as I mentioned earlier, our SDA kit enables a variety of different multi-omics add-ons to the standard 3' single cell sequencing that PIPseq is based on. So this includes, as I've already shown examples in the slide deck of using antibody-based oligos or SNTs to get additional information about surface epitope expression, as well as sample multiplexing. Also, looking into genetic perturbations via CropSeq. Fortunately, we had a few users ask about this. We are unfortunately not compatible at the moment with ATAC-seq or other epigenetic labeling approaches, in addition to compared with gene expression. That is, however, on our product roadmap, although a little bit further out than some of the other upcoming releases that I mentioned at the end of the talk.
Cool. And can you talk about the bioinformatics platform a little bit more in detail? I mean, what does it do, and where do you need to kind of move from the PIPseq bioinformatics to some kind of other analysis tools?
Yeah, so for those users that have run other single cell platforms before, you'll find the experience to be highly similar. You just need to input your FASTQ files that come off of the sequencer, as well as a reference directory to their genome that you're trying to map to, and then the software will output a report as well as the gene expression matrix that you can take into your own tertiary analyses. There's a variety of other settings that you can explore if you look at the user manual for PIPseq that you can use to tune the output. But at the very base, that's kind of some of the main core outputs of the bioinformatic pipeline.
Cool. And what about samples, particularly fixed samples?
Yeah, so while we are compatible with fixation, it's unfortunately limited at this point to DSP methanol-based fixatives. So this means that we won't be able to, we don't have a solution at the moment to analyze, for example, archived samples that have been fixed in PFA and stored for tens of years. That is something that is on our product development roadmap, but again, it's at a later stage or earlier stage, I guess, in the product development process. So the fixation, that I mentioned is really a tool to be able to, if you are planning on running a time course study or you're asynchronously obtaining precious clinical samples, you may choose to take advantage of a fixation so that when you're ready, you can actually start all of your PIPseqs at the same time.
I have one more point to make. I did a presentation last week at WeHi, and we were talking about the workflow. In comparison to other platforms, Jesse, can you talk about the workflow?
Yeah, so I would say that those of you, again, not to name, well, I'll name some specific comparisons here, but those of you that are familiar with 10x, you'll find the workflow to be highly similar, about the same length. And those of you who tried other split pool-based approaches, such as parse or scale kits, the workflow involves much less pipetting. In fact, in contrast to kind of intuition, where you think if you scale up to more cells, your workflow will get more complicated, what we found is generally users actually like the ease of use of our larger volume kits. In fact, the T20 seems to be quite easy to handle relative. People really like the workflow of the T20 assay. So I think that's also a great starting point for any of you that are looking to give PIPseq a try.
Fabulous. Okay, that's all the questions that I have. Oh, hang on, there's one question in Q&A. Thomas has said the scalability is excellent. He'll be reading into this and seeing how we can implement this in our lab. Thanks, Tom. We will definitely be in touch in terms of how we can implement it. If you would like to have a specific, actually, I'll be in touch to see if there's any specific answers that we can actually answer with this, or questions that we can answer with this system.
Yeah, and then I'll be around after the seminar. So if you want to schedule a follow-up conversation about how best to apply PIPseq to your particular project, I have, before I started working on the international commercialization of PIPseq, I spent 3 years really within the R&D team building the product. So I definitely have quite a bit of familiarity with the assay. Happy to kind of work with Catherine to facilitate a time to do a follow-up one-on-one meeting.
Sure. Just to give you some idea, I think there's only a few people left, but just to give you some idea with in terms of pricing, I did a quote the other day for a customer for 64 samples. It came to about 120,000 Australian dollars. The comparative quote from another competitive product was in the order of $500,000. So in terms of scalability and pricing, it's certainly definitely a lot better than what's currently on the market. So I'll leave that with you guys. Thank you very much for your time. And as I said, we'll be recording this for anybody who's missed it. And if you have any further questions, please reach out to